New research by investigators at Brigham and Women's Hospital and Rockefeller University helps address a long-standing question about the inflammatory skin condition psoriasis. Why do skin lesions that have resolved with therapy recur in the same locations after a patient stops using topical steroids? According to BWH physician scientist Rachel Clark, MD, PhD, of the Department of Dermatology, researchers have been searching for years for a cell population that continues to smolder after psoriasis appears to resolve. It's been challenging to zoom in on the population of T cells driving the disease, in part because when psoriasis is active, lesions are flooded with diverse T cells. But Clark and her colleagues have taken a new approach. Instead of looking during the height of activity, they examined lesion sites after treatment and identified T-cell receptors of cells at these sites that were shared across psoriatic patients, but not found in healthy individuals or those with other skin conditions. The team's findings are reported in the Journal of Clinical Investigation. When psoriasis is treated, T-cells that flooded in during inflammation recede like the tide, said Clark, who is the corresponding author of the paper. They leave behind a population of cells that stand out. The population of T cells that remains are tissue-resident memory cells, which live long-term in skin and, when functioning properly, should be fighting infection. But for patients with psoriasis, these cells may be the source of the misguided immune response that leads to red, inflamed patches on the skin. To identify this T-cell population, the researchers took biopsies at the sites of active lesions before treatment and biopsies of the same skin areas after the lesions had cleared on therapy. Using high-throughput sequencing and immunostaining, the research team found that resolved lesions contain populations of T cells derived from just a few cells known as oligoclonal populations that produced IL-17, a telltale marker of inflammation. These cells also shared stretches of genetic sequence that code for the same antigen receptors. These shared T-cell antigen receptors were found only among cells from psoriatic patients, not in cells from healthy controls or people with skin conditions such as atopic dermatitis. This work highlights the fact that most psoriasis treatments do not kill these disease-causing T cells, but instead temporarily suppress their activation. Now that they have identified the long-lived, skin-resident T-cell population that appears to be driving recurrence, the team plans to search for new therapies that can deplete these resident T-cells, potentially driving the disease into long-term remission. We believe these resident memory T-cells are the root of the problem. Imagine these cells are teenagers throwing a party. They invite lots of other cells to the site of the party, making it hard to identify them. While the party is in full swing, it's only after inflammation dies down and everyone else goes home that we can see these culprits, said Clark. A small number of cells can cause so much trouble. But depleting this population of cells may be the key to slowing down this disease or preventing its recurrence. This work was supported by the National Institute of Arthritis and Musculoskeletal and Skin Diseases NIAMS R01AR063962, NIAMS R01AR056720, NIAMS P30AR069625, National Institute of Allergy and Infectious Diseases NIAID R01AI097128, R01CA203721, T32AR070986, the National Psoriasis Foundation, the American Skin Association, and an AstraZeneca Foundation Faculty of Medicine of the University of Lisbon research grant. Paper cited Modest TR et al. Clinically resolved psoriatic lesions contain psoriasis-specific IL-17 producing plus or minus superscript 2 T-cell clones JCI DOI 10.1172 JCI 93396. Disclaimer: Autism and uric alert are not responsible for the accuracy of news releases posted to Uric Alert by contributing institutions, or for the use of any information through the Uric Alert system.